Good afternoon, everybody. I'm coming at you recorded today. It's unfortunate I can't be there with you all, so bear with me. I hope you all can hear me. I'm excited to talk today about a very large team effort related to our CSMI work on Lake Huron, which we kicked off in 2022. I'm going to jump right in. First, I'd like to acknowledge a lot of folks that helped out with this work. So many folks helped in the field and laboratory. I also want to acknowledge our our funding sources, GLRI and the Corriganine Restoration Framework, and a lot of folks that helped with collaboration and data sharing, and also just sharing methods for larval fish sampling over the years. So big thanks to all of you. I'm just going to start right in on the problem. There's plenty of evidence on Lake Huron that lake whitefish are facing a recruitment bottleneck. And we hypothesize it's in the early stages of their life, potentially in the first year of their life. And so a recruitment bottleneck is just a way to say that something is killing them in their first year of life. They're not really making it in high numbers to that catchable size. And so the core of this work is investigating what factor or more likely many factors are killing these fish. I'm going to be mentioning a lot of terms about early life history for lake whitefish and developmental stages, so I think it'll be helpful just to show a figure of the life cycle quickly to explain those. So whitefish deposit their eggs in the fall, and those incubate through the winter. They hatch in the spring, and these are considered yolk sac larvae. They're very small, recently hatched. They have a yolk sac on their body, and typically they come to the surface. In Lake Huron, they're known to be at the surface and near beaches in nearshore zones where it's kind of like nursery areas for them. Early to mid-spring, they absorb that yolk sac. These are what I'll call post-yolk larvae, still larval fish. As they grow into the summer, I'll call these post-larval juveniles. Sorry, that's cut off there. These are age 0 juveniles, essentially. And then as they grow through multiple seasons, you have age 1 and 2 juveniles. And then age 3 plus, you have those adults. And the cycle starts again. So the overall goal of this work is, can we partner up with other agencies and monitor a broad range of sites across Lake Huron, the widest scope possible, to reveal these variables that may be responsible for that low lake whitefish survival at that early life stage. We have a couple of research questions that I'll check in with here and then also follow up with at the end of the talk. First, we predicted that we have this north to south design in the study where we monitored northern Lake Huron sites and southern Lake Huron sites because there's this difference in nutrient gradient between those regions. In the North Channel and northern Lake Huron, it tends to be more nutrient rich. Dreissenid mussels, those are invasive mussels, they haven't quite established as well up there. And so the lower food web is just booming a little bit more up there. In the south, like south of the thumb of Lake Huron, the water is very crystal clear. Those Dreissenid mussels are really heavily established. And so things are much different down there in terms of nutrients. So we predicted, one, that larval lake whitefish densities would be greater in the north versus the south because of that higher nutrients. We also predicted that greater zooplankton densities would be found in the north versus the south. Following all that, we predicted better larval lake whitefish growth in the north versus the south, and you can look at that by length at age relationships, which I'll talk about with larvae. We then predicted for those post-larval age 0 fish that there'd be more of those found up north in our nets compared to down south. Again, just related to the nutrients. And then to tie it all together, our fifth research question is that since there's less food available down south, that means less food in their bellies, and that is, what is responsible for that slower growth if we see it. So to clarify the methods, I'll also show some figures here to talk about what we did. In the early spring, right at ice out, we sampled on beaches with hand-towed gear. We're towing it by hand. We're wading in the beach. These are Newston nets. They're larval fish nets. And at 4 of the 8 sites, we also used small vessels and towed larval fish nets just out from the same beach. This way we could compare beach densities to those densities just off of the same beach, a very local scale comparison, just to see how many actually live on the beach in that beach zone. In these early days, we collected larval fish and zooplankton with a zooplankton net alongside all those larval fish tows. Then as summer kicked off, these fish are growing up. We went to continue to catch them through their first year of life, essentially. And so we were seining, beach seining for those post-larval age 0 juveniles, and also still taking those zooplankton samples alongside those hauls. So to start with the early ice out larval work, we collected larval fish and zooplankton. We identified and counted all of those. And for the fish, we did genetics to identify them further because you need genetics for these larval carrageenans. We did some morphometrics on the larvae, aging, and diets. Just a quick snapshot of the sites using a map here. There are the 4 North Channel beaches we sampled. And then down south from Harbor Beach to Port Huron, those are the 4 southern sites I'll talk about. I'll just quick do a spoiler alert on some of the big stuff we found. One is that we found an order of magnitude more larval whitefish right on the beach compared to just off of the same beaches, which emphasizes this sort of shouldn't surprise the larval folks in the room. And according to the literature, the beaches are very important for these fish. This just kind of confirmed how important those beaches can be. A lot of larval lake whitefish use that beach zone, that less than a meter deep, warm beach zone in the early spring like that. So that was interesting. The same beach trend we saw with zooplankton, orders of magnitude more zooplankton right on the beach compared to just off the beach. North to south, we saw orders of magnitude more zooplankton up north compared to down south as well. So there's just more nutrients, perhaps that translates to more zooplankton up north. And larval lake whitefish densities across those regions were really variable. So I'll expand on that part a little more. Here's a look at overall larval fish densities at those sites. North Channel is the light blue and Southern Lake Huron is the peach color. And you'll see this is just mean density overall of lake whitefish. It's lake whitefish per cubic meter. You'll see right away that there's this peak in density at Harbor Beach down south, and this surprised us. Compared to the North Channel sites, it actually was significantly greater down south. So that went against one of our predictions that we'd see more higher densities up north. If I take that same larval fish graph I just showed and overlay it on our zooplankton, our overall zooplankton densities, just number of zooplankton per liter, I just wanted to quickly point out that having very low whitefish densities doesn't necessarily mean you'd have low zooplankton densities at those sites, and vice versa. In some cases, we had really high zooplankton densities and extremely low lake whitefish densities. And in other sites, very high lake whitefish larvae densities and extremely low zooplankton, like Harbor Beach here. So that might not surprise folks either. I just think that that's a really important overall thing that we saw. We need to unpack this further. For example, we need to look at succession of zooplankton across these beaches. What is hatching first and what is available to eat? What are these fish eating as they live their several weeks on the beach? What can they fit in their mouth versus what is present? That kind of thing we're still sort of unwrapping. If I just choose one site in the north and one site in the south to compare for sake of time here, I'll choose these 2 sites and expand on this a little more. So the North Channel site is Fourth Sand Beach, and these are just the larval fish densities across weeks that we sampled. So they're unraveled a little bit more here. And you see generally at Fourth Sand Beach, the larvae were pretty low densities on the beach, and then they peaked around 11 May at around a fish per cubic meter, a little lower than that, but they hit about a fish per cubic meter. We saw similar densities at Harbor Beach and similar trend. And so that's why we selected these 2 sites to prioritize first for further analyses. If you just look at the length of the fish we caught each week, you'll see that at Fourth Sand Beach up north, they continued to get, they were bigger fish as we sampled each week. Down south, they never really got any bigger each week. And again, this could be explained by a lot of different things. Perhaps down south, the fish were cycling through more. It wasn't the same population, fish were moving, but it certainly generated some research questions for us. So we dug in further. Here I'll just show the length at ages for each of those sites. So Fourth Sand Beach up north will be in blue. Harbor Beach down south will be in orange. And this is just the length of fish versus the estimated age that we aged them with their otoliths. And I'll show yolk sac larvae first, then add post-yolk larvae, and then show everything together because they tend to be a little different. They're at very different developmental stages. So as you can see, up north, Fourth Sand Beach, as the fish were getting longer, they were getting older. It's a little messy, but it generally, shows that trend. If you add Harbor Beach fish, they pretty much overlap. This isn't too surprising since they're yolk sac fish. They have basically a little battery pack of nutrients on them at this stage. So they're not, we predicted that they wouldn't be stunted at this stage per se, but it was worth looking closer at. With the post-yolk larvae, again, a little messy, but they still keep getting older as they get longer. Adding the larval fish from Harbor Beach down south, though, you can see they're starting to fall below the curve now. So they are getting longer as they're aging, but they're below the curve of the North Channel fish, suggesting they might be stunted. Adding all the larvae together, you see that they do indeed start to fall below the curve as these larvae are growing up down south versus the larvae growing up north. So that's with us aging the otoliths. We also just took the size of the otolith, which is another good proxy for age. Otoliths tend to grow as a body grows in sort of a uniform way, unless a fish is stunted. So size of otolith can be a good proxy for age. So again, the relationship with yolk sac fish up north, the otolith was getting larger as the fish was getting longer. It's kind of messy, but it's generally linear there. And right away, even as yolk sac fish, the Harbor Beach fish were falling below the curve, meaning that their otoliths were growing in their head, but their bodies weren't getting as long as the North Channel fish, even at this early yolk sac stage. Post-yolk larvae, you see the trend. They continue to fall below the curve in terms of size of the otolith versus length of the body down south. So as you add everything together, you see that, again, Harbor Beach is below the curve in terms of length. A different way to look at this is just otolith max diameter as a proportion of the total length. And what you'll see right away in the yolk sac fish on the far left is that the proportion being larger in Harbor Beach fish means their body was smaller in proportion to their otolith compared to North Channel fish. So they seem to be stunted, and it's driven, it's starting out in this early yolk sac stage. Here's a quick picture of a larval otolith. I just want to show another thing that we measured. So you see, it's kind of hard to see. It looks kind of like a tree stump. You see these daily rings. Some of them appear to be a little thicker. In the literature, these are called check marks. That's essentially a day or two when growth slowed down. And we counted those up because those can indicate a stressful event for the fish. So they happen when a fish hatches, when they absorb their yolk sac, if they get a cold shock, that kind of thing. So we counted these up as a proxy for stressful events. And if you look at the yolk sac fish, when we count up the checks per otolith, Harbor Beach down south had way more variable check marks on their otoliths compared to the North Channel fish, suggesting that possibly they're going through more stressful events or their stress is more variable down south. Checking in on seining, this is all underway right now in terms of analyzing these seine data. But essentially we continued sampling, caught juvenile age 0 fish, zooplankton, counted everything, identified stuff. We got together with multiple agencies that are listed on this talk U.S. Fish and Wildlife Service, Ontario Ministry of Natural Resources. We're combining our data sets and looking at our aging and diets across these fish and regions. A quick snapshot of the densities that we have mapped so far. You can see that it's all over the place. Even more variable than the larvae, it seems. The peak densities were actually near Alpena, and up north and near the fishing islands, things were a little lower. Down south, where we got all those larvae, we age 0 juveniles. We caught none in our seines, and that's really interesting. We don't know where those fish went. We're beginning to work on diets. This is underway. I just wanted to quickly say that the diets are really variable across regions, across years. This will be really interesting to unpack. So checking back in on our research questions. Greater larval fish densities north versus south? Not necessarily. We saw Harbor Beach had this peak in larval whitefish densities. Greater zooplankton densities in the north versus south? Yes, that is occurring. There seems to be way more zooplankton up north so far in our analyses. Better larval lake whitefish growth in the north versus the south? So far, yes, with the sites that we've analyzed, but we have more work to do there. Did we see greater post-larval age 0 densities in the north versus the south? No, not necessarily. That was the peak densities were near Alpena that we saw, and they seem to be all over the place. So more work needs to be done there. The less food in this available means less food in stomachs question and slower growth. We're well underway working on this one, so I'm looking forward to updating the group on this when we get it more developed. So that's the scoop with CSMI work on Lake Huron and investigating these recruitment constraints. I have my email there. Again, I'm sorry I can't be there in person, but please reach out via email or send me a text if this generated some thoughts from you, or if you're sampling larval fish or juvenile whitefish and you want to get together and talk. I'd be really interested in that. Thanks so much for listening, and have a great rest of the conference.